Okay, so welcome everybody, and thanks a lot to Philip for inviting me to give this talk today online to you. So my lab is interested in how metabolic rewiring is supporting metastasizing cancer cells. And as you all know, metabolic rewiring is a hallmark of cancer cells in general. But you may now wonder actually, why do we think that metabolic rewiring is so important for metastasizing cancer cells? And the reason for this is actually twofold. So first of all, the cancer cells that undergo metastasis formation need to change their phenotype. So basically, when you look at a primary tumor and an established metastasis, most cancer cells within these two tumor entities are in a kind of proliferative mode. But proliferation becomes less important once the cancer cells metastasize, because first they need to invade the surrounding tissue, they basically need to survive in the circulation, then they colonize a distant organ, and only then they slowly get back into this kind of proliferative phenotype. So clearly, cancer cells that successfully metastasize need to change their phenotype. Moreover, we cannot necessarily assume that cancer cells have access to the same nutrients when growing as a metastasis or a primary tumor, because they're simply growing in different organs, and these organs may provide different nutrients to the cancer cells. So since metabolism is now the biological process that connects the nutrient microenvironment to the cellular phenotype, we actually believe it is very important to understand the metabolic rewiring that occurs during metastasis formation to inhibit the ability of the cancer cells to change their phenotype and therefore inhibit their ability to successfully metastasize to distant organs. And the validity of the concept that the nutrients are so important, we have shown quite a while ago in a first study where we focused on pyruvate, because we actually found that pyruvate was enriched in the lung environment, which is a frequent metastatic site of many cancers, including breast cancers. Moreover, we found that the cancer cells that reach the lung environment, they actually will adapt to the pyruvate availability. So clearly, pyruvate was shaping the metabolism of the arising metastasis. But obviously, we wanted to know whether pyruvate only shapes the metabolism of the arising metastasis, or whether it is really required for effective metastatic outgrowth. So to address this question, we decided to inhibit the ability of the cancer cells to actually take up pyruvate from the extracellular space. And we did this by deleting the pyruvate transporter MCT2. If you delete this transporter, the cancer cells cannot take up any longer pyruvate from the extracellular space, but they can still make their own pyruvate. So we recreated the MCT2 deletion and parental cells, and we injected them into the memory fat pad of mice, and then we monitored primary tumor growth and the arising metastasis. So when we looked at primary tumor growth, we actually did not see any difference between the control cells and the MCT2 deleted cancer cells, in line of the idea that pyruvate is not an important nutrient in the memory fat pad where the primary tumors are growing. However, when we then started looking into the lungs, we made quite a significant observation, because we could clearly see that in the lungs, the MCT2 deleted cancer cells had really a hard time growing out, so a hard time building metastasis. So that was really quite exciting, because this was now really showing that pyruvate not only shapes the metabolism of the rising metastasis, but it is really required for effective metastatic outgrowth. So we obviously wanted to know, why is this the case? So why is pyruvate so important for the metastasizing cancer cells? And so we thought to define this mechanism, it would be actually good to have an in vitro system that recapitulates our in vivo finding. And to define such, we went back to a study we had concluded a couple of years back, where we had actually found that when we put breast cancer cells in 2D culture, they were more reflective of the metabolism of primary breast cancers, while when we put the same breast cancer cells in 3D culture, this was actually more reflective of the metabolism of the arising metastasis. So therefore, we hypothesized that we could use 3D versus 2D culture to actually mimic the response of metastasis versus primary tumors to pyruvate availability. And indeed, this was possible because when we withdraw pyruvate from the 2D cultures, there was a slight decrease in proliferation, but overall the cells were doing fine. But when we did the same, the pyruvate withdraw in 3D culture, clearly the colonies could not form anymore. So that was great because now we had an in vitro system recapitulating our in vivo finding. But we thought maybe the fact that we can recapitulate this observation from metastasis versus primary tumors in 3D versus 2D culture already tells us something about the mechanism we are looking for. Because a major difference between 2D and 3D cultures is actually that in the 2D cultures, the cancer cells adhere to a pre-existing matrix. So they immediately get growth and survival signaling. But when you actually look into the 3D cultures, the cancer cells actually cannot adhere to a pre-existing matrix because you either put them on ultra low attachment plates or you put them on something like soft agar. So they first need to make their own extracellular matrix to basically get growth and survival signaling. So based on this, we hypothesized may it be actually possible that pyruvate is important for extracellular matrix remodeling. So to test this, what we simply did is we gave extracellular matrix to the 3D cultured cancer cells, and we asked, do they now grow without pyruvate? And indeed, when we did this, we could clearly see that without pyruvate, there's hardly any colony formation. But when you then give marticel, which is a form of extracellular matrix, and you still don't have pyruvate in the media, the colonies can form again. If you do the same experiment, so this marticel addition in the presence of pyruvate, the pyruvate uptake of the cancer cells is actually decreasing. So clearly highlighting that extracellular matrix metabolism actually depends on pyruvate availability, at least in 3D cultures. So that was interesting, but is extracellular matrix metabolism now really important for metastasis formation? So luckily other people had already looked into that, and it is very clear that cancer cells that metastasize to the lung environment, they need to deposit their own extracellular matrix in the form of collagen to promote metastatic outgrowth. And it was further found that one of the rate-limiting enzymes that allows the cancer cells to do this is collagen proline 4-hydroxylase. So this enzyme actually hydroxylates the proline residues of the pro-collagen molecule, and only a sufficiently hydroxylated pro-collagen molecule will actually be able to form a stable triple helix of collagen, which in turn is functional in supporting growth and survival signaling. So therefore, we actually hypothesized, may it be possible, that pyruvate is important for the activity of this enzyme, collagen proline 4-hydroxylase. To test this, we simply measured collagen hydroxylation. And when we did this in different breast cancer cell lines, we actually could clearly see that whenever we gave pyruvate, we could really boost collagen hydroxylation. So that was exciting because clearly pyruvate was linked to collagen hydroxylation. But the question was how? So what is happening in between? And so to define this mechanistic link between pyruvate and collagen hydroxylation, we decided to do a metabolomics approach with the rationale that the metabolites that change most upon pyruvate withdrawal could actually link pyruvate to collagen hydroxylation. And when we did this, we could see that besides pyruvate, alpha-ketoglutarate, and to a minor extent malate and citrate decreased in abundance. So we hypothesized that one of these metabolites could link pyruvate to collagen hydroxylation. To test which of these metabolites is now important, what we did is we supplemented them back into the media and measured collagen hydroxylation. And when we did this, we could clearly see that only one metabolite, namely alpha-ketoglutarate, was able to drive collagen hydroxylation to the same level than pyruvate. And this actually makes a lot of sense because alpha-ketoglutarate is the co-substrate of the collagen proline 4-hydroxylase reaction. Because in this reaction, alpha-ketoglutarate and proline are converted to succinate and hydroxyproline. So which basically means that the higher the alpha-ketoglutarate concentrations, the more active will be collagen proline 4-hydroxylase because you have a metabolic regulation of the enzyme. So that was quite exciting, but you may now ask another question, and that is, what about the known transcriptional regulation of collagen proline 4-hydroxylase? Because this enzyme is known to be highly transcriptionally regulated. So we also wondered about this, and we decided to overexpress collagen proline 4 hydroxylase and ask, do we now still need pyruvate to activate collagen hydroxylation, or is the overexpression sufficient? So when we did this, we could clearly see that despite the fact that the overexpression of collagen proline 4 hydroxylase slightly increased collagen hydroxylation, you were even in the overexpression cells, you were either needing pyruvate or alpha-ketoglutarate to drive collagen hydroxylation. So that was actually really exciting, because this makes clear that besides the known transcriptional regulation, we definitely also need the metabolic regulation of this important process. So before going on, we thought there is one remaining small question, and that is, how does actually pyruvate increase alpha-ketoglutarate levels? And we thought probably it's very simple. The pyruvate goes to the mitochondria, it can fuel the TCA cycle oxidatively and anaplerotically, and this leads to alpha-ketoglutarate production. But we wanted to be sure about that. So we said, let's do a pyruvate tracing and let's see, do we really find carbons of the pyruvate in alpha-ketoglutarate? But when we did this, this was actually a quite stunning result, because differently to our expectation, we actually did not see much label of pyruvate in alpha-ketoglutarate. So we therefore started wondering, where does the pyruvate carbon go? And when we looked in our mass spectrometry data, we actually found a lot of the labeling goes to alanine. And in line with this observation, we actually saw that whenever we gave pyruvate to the cancer cells, they actually started secreting alanine. So we thought, how can we actually reconcile this data? And we looked at the metabolic network, and we realized we can reconcile all these observations when we have activity of alanine aminotransferase. Because in this reaction, pyruvate and glutamate are converted to alanine and alpha-ketoglutarate, and the transfer is actually a nitrogen transfer, and the carbon is going to alanine. So to actually verify that this is indeed the case, we decided to silence the alanine aminotransferase and measure alpha-ketoglutarate levels. And indeed, when we did this, we could clearly see alpha-ketoglutarate levels dropped up in alanine aminotransferase silencing, and accordingly also the levels of hydroxylated collagen. So based on this, we can actually conclude that pyruvate from the extracellular space is transported via MCT2 into the cells. The pyruvate is then fueling alpha-ketoglutarate production via the alanine aminotransferase reaction, and this alpha-ketoglutarate is metabolically activating collagen proline 4 hydroxylase to basically generate hydroxylated procollagen. But the important question now is this whole process of functional relevance? So in other words, is the collagen that is produced in the presence of pyruvate now really of greater stability? So we decided to test this, and we decided to test this with an MMP assay. And the background of this assay is that if you have collagen of lower or higher stability, matrix metalloproteinase will be actually differently suited to digest this collagen, because lower stability collagen, the MMP is more efficient to digest it. So you see actually more hydroxyproline in the supernatant of the cells. So we therefore actually let the cells produce collagen in the absence or presence of pyruvate, and then we digested it with an MMP to look how much hydroxyproline we see in the supernatant. And when we did it, it was actually very clear that in the absence of pyruvate, much more hydroxyproline was in the supernatant, which basically means the collagen that was produced was of low stability, and we actually could be rescuing this collagen stability when giving alpha-ketoglutarate. And this was not only true in the in vitro system, but also in vivo, in the mice, because when we gave the mice an MCT2 inhibitor, we actually saw that cancer cells in the metastatic niche were less able to hydroxylate collagen, and also the deposition of stable collagen 1 and 3 was actually reduced up in an MCT inhibitor treatment in the metastatic niche. Moreover, we could also verify this link to alpha-ketoglutarate, because when we gave the mice the MCT2 inhibitor, but at the same time alpha-ketoglutarate, we rescued collagen hydroxylation in the metastatic niche and also the deposition of collagen 1 and free. So this actually leads to this result that pyruvate from the extracellular space is really important to metabolically drive collagen hydroxylation, and by creating a permissive extracellular matrix environment, this supports metastatic outgrowth. In the consequence, if you then actually silence or inhibit MCT2, you can inhibit metastatic growth. But this observation that this metastasizing cancer cells depend so much on their own extracellular matrix formation was actually raising a next question, and that was, where is the ATP, so the energy coming from, that the cancer cells actually need to traffic all this collagen? Because protein synthesis and trafficking is one of the most energy consuming processes in a cell. So we also wondered about that, and we decided to actually quantify the activity of metabolic pathways that can produce ATP in 3D culture versus 2D culture, actually utilizing them as a mimic for metastasis formation versus primary tumor growth. And so what we did is we fed this 3D versus 2D cultured cancer cells labeled glucose or labeled glutamine. And then we looked which metabolic pathways are active. And when we did this, we made quite a surprising observation, because we saw the greatest change between 3D and 2D culture was in the production of proline. Because what we actually found was that it was less glucose and less glutamine contribution to proline production. And what does this now mean? This basically means that also in 3D culture, some glucose and some glutamine was used to make proline, but there was, compared to 2D culture, an increased fraction of proline that was neither labeled or neither coming from glucose and glutamine. And we hypothesized that this was coming from proline uptake. And indeed, when we measured proline uptake, it became very evident that while proline is secreted in 2D culture, the cancer cells start taking up the proline in 3D culture. And so when we then looked at enzymes related to proline metabolism, we could actually see that one enzyme was highly expressed in 3D culture, and that was ProtDH. So what is ProtDH doing? So ProtDH is an enzyme that catabolizes actually proline. And what is so interesting about this is that this is coupled to ATP production in the mitochondria. So to test now whether indeed cancer cells in 3D culture rely on ProtDH activity to produce ATP, we decided to do a dynamic metabolomics experiment. So what we basically did is we acutely inhibited ProtDH and look, do ATP levels drop? And indeed, when we did this, we could nicely see that once we give the ProtDH inhibitors, ATP levels dropped over time. So clearly indicating that proline catabolism or ProtDH activity is important for ATP production in the 3D cultured breast cancer cells. But we also wanted to be really sure about that. So we said, let's take a closer look on how ProtDH activity is actually leading to ATP production. And when you look at the metabolic network, then it's very clear that the electrons generated in the ProtDH reaction are actually funneled into complex 2. So basically this means that the ATP production from proline catabolism depends on complex 2, 3, 5, and 4 complex activity, but actually bypasses complex 1. So in the consequence, this should actually mean if really ProtDH activity is important for energy production, we should see that the cancer cells are sensitive to antimycin, but that they're actually insensitive to rotenone, which inhibits complex 1. So indeed, when we tested this, we could see the expected phenotype that the cancer cells did not care in 3D culture about rotenone, but they greatly reduced colony formation once we inhibited complex 3, which means this is consistent with the idea that proline catabolism is important for energy production. The next question we now addressed was, is it actually sufficient to inhibit ProtDH to reduce colony formation? And indeed, when we do this in vitro, we can nicely see that when we use a cancer cells, we give a protease inhibitor, we actually reduce colony formation. And I think it's important to note, if we do the same thing in a memory epithelial cells, so non-transformed cells, they actually do not care about the protease inhibitor. So the final experiment, what we wanted to do is, we wanted to check, is this also working in vivo? So can we reduce metastasis formation by giving LTHFA, so by inhibiting protease? And so we did a dose response in the mice, and indeed what you can see here is the higher the dose of the protease inhibitor, the more we reduce metastasis formation. So this actually means that there is quite an intertwined need of pyruvate and proline in cancer cells that undergo metastatic colonization, because the pyruvate actually drives the ability of the cancer cells to hydroxylate proline of the pro-collagen molecule. And note that collagen is a proline-rich protein. And at the same time, this whole collagen production and trafficking to the extracellular space is energy consuming, which the cancer cells, at least in part, fuel by proline catabolism. So this actually nicely tells us that there's a certain metabolic need for cancer cells that have this phenotype of metastatic colonization. But obviously, to grow into an advanced metastasis, cancer cells at a certain moment also need to activate growth signaling. And so we actually started wondering whether cancer cells in the metastasis or the primary tumor use different growth signaling pathways, or if they rely on the same growth signaling pathways. So we decided to address this question by a very simplistic approach, because we just took a mouse that had a primary breast tumor and lung metastasis, we isolated tissue from both, and then we looked what is the activity of major growth signaling pathways, and predominantly we looked at the MTOR complex 1 pathway, which is one of the most known growth signaling pathways. And we quantified the activity of the MTOR complex 1 pathway based on phosphorylation of its downstream targets. And when we did this, we actually could clearly see that both in the primary tumor and the metastasis, we have phosphorylation of mTOR targets, which actually means that both primary tumor cells and metastasis cells do use mTOR complex 1 signaling. So why this looks not so exciting at the first glance, we got actually quite excited about this when looking at several of these Western blots, looking at different mice, because we started realizing a pattern. And this pattern was that it seemed to be the case that we had always higher mTOR target phosphorylation in the metastasis compared to the primary tumor. And this was not only true for the mice, but also for patient data, because when we looked at paired samples from breast cancer, primary tumors, and metastasis, we saw that in 4 out of 5 patients, we had higher mTOR activity in the metastasis compared to the primary tumor. So based on this observation, we hypothesized that yes, indeed, both primary tumor and metastasis use mTOR complex 1 signaling, but they actually may activate it by different means, which could explain the differential magnitude of activity we observed in metastasis versus primary tumor. So to actually go further and understand this better, we said we need to know how is mTOR activated. And as you all know, mTOR has different means to be activated through growth factors, insulin, but actually also through nutrients. And since we already know that the nutrient microenvironment is different between the primary tumor and the metastasis, we decided to focus on nutrients. And what we did is we actually correlated nutrient transporter expression with mTOR activity in breast cancer samples. And when we did this, we saw that out of the tested nutrient transporters, 2 of them actually nicely correlated with mTOR activity. And that was the expression of the glucose transporter GLUT1, and again, the expression of the pyruvate transporter MCT2. So both of these transporters could now be linked to mTOR activity in the metastasis, but not in the primary tumors. So to actually decide on which transporter we should focus, we thought, let's see which nutrients are predominantly available in the lungs versus the blood plasma. And as I have told you already in the beginning of the talk, we know that pyruvate is actually enriched in the lung environment, because when we look at lung interstitial fluid, which is the first source of nutrients metastasizing cancer cells will see, and we compare this to blood plasma, we actually see that pyruvate is enriched. We did the same analysis for glucose, and when we do this, we actually see that glucose availability is actually reduced compared to blood in lung interstitial fluid. So we decided to focus on pyruvate because it was enriched in the lung. And so the hypothesis was that pyruvate uptake would help to activate mTOR complex 1 signaling. So if this is the case, what we actually would expect is that if we acutely inhibit pyruvate uptake, we should see a response in mTOR complex 1 signaling in the metastasis, but not in the primary tumors. So we did this by, again, having a mouse with a primary tumor and metastasis. We acutely dosed the mouse with an MCT2 inhibitor, and then we looked at phosphorylation of mTOR targets. And when we looked into the primary tumor, we could actually clearly see that there was no difference, because the phosphorylation of mTOR targets was exactly the same, regardless if we gave the MCT2 inhibitor or not. But this actually looked quite different when we went into the lung metastasis, because there we clearly could see that the phosphorylation of mTOR targets really decreased once we gave the MCT2 inhibitor. So this actually would indeed suggest that pyruvate is important to potentiate mTOR complex 1 signaling in the lung metastasis. But the question now is, how can pyruvate now have an impact on mTOR complex 1 signaling? And so to define this mechanism, we went back to the in vitro system. We used 2D cultures because they are better reflective of the metabolism of proliferating cancer cells. And we gave an increasing amount of pyruvate to these 2D cultures. And then we looked which metabolites are changing. And we were actually quite excited to see that the 5 highest ranking metabolites that were changing up in pyruvate addition, 4 of those we could actually link via one pathway, and that is the serine biosynthesis pathway. Because as the name says, the serine biosynthesis pathway makes serine from free phosphoglycerate, a metabolite of glycolysis. It has been shown previously that the pathway is activated by NAD availability. And what we found very interesting, the pathway makes equimolar amounts of alpha-ketoglutarate. And that was so interesting to us because alpha-ketoglutarate has actually been shown to activate mTOR complex 1 signaling. So we hypothesized that a pyruvate-enriched environment would drive serine biosynthesis. But if this is true, this should also happen actually in vivo, in the metastasis compared to the primary tumor, because we know the metastases are in a pyruvate-enriched environment. So we checked this, and indeed we saw the metastasis had a higher serine biosynthesis, increased alpha-ketoglutarate and free phosphoglycerate levels, and also an elevated NAD to NADH ratio. So clearly, the environment of the metastasis, so the lung environment, was promoting the serine biosynthesis pathway. But the important question now is this now really modulating mTOR activity? So to test this, we decided to inhibit the serine biosynthesis pathway by silencing its first and important enzyme, PHDDH. If you silence this enzyme, the cells cannot generate de novo serine. So this will be decreased serine de novo production, decreased cytosolic alpha-ketoglutarate production. But since actually the cells in vitro are in a serine-enriched environment, and also the lung has serine, they still can fuel actually the serine they need for protein synthesis. So then we basically use the PHDDH silence cancer cells, and we ask, does PHDDH silencing change phosphorylation of mTOR targets? First, we again looked into the primary tumor, and in line with the overall hypothesis, we saw no changes in phosphorylation between PHDDH silencing and controls in the primary tumor. But when we then again looked at the lung metastasis, we could actually clearly see that in the lung metastasis, phosphorylation of mTOR targets was actually decreased. So that was actually really quite exciting, because this would really suggest that pyruvate from the extracellular space is also important in proliferating cancer cells, because it drives the serine biosynthesis pathway, which will help them to activate mTOR complex 1 signaling. But you may now argue, well, all what I have shown you so far are phosphorylation Western blots. So what about really functional relevance of these changes in phosphorylation? So we also wondered about that. And so what we did is, to show functional relevance, is we actually treated the mice with rapamycin, which is an mTOR inhibitor. Because you actually would expect that the higher the mTOR activity in a cancer cell, the more responsive it is to rapamycin treatment. And the first thing what we did is we looked into the primary tumor. And in line with the idea that these primary tumors actually do use mTOR complex 1 signaling, we actually see that they are somewhat sensitive to rapamycin. The next thing what we did is we silenced PHGDH and again looked at rapamycin sensitivity. From our phosphorylation data, we would expect that this should not have any impact on rapamycin sensitivity, because we didn't see any changes in phosphorylation. And this is what we also observed. So clearly the cancers, the primary cancers, are similarly sensitive to rapamycin, regardless if they have PHGDH or not. Then we did the same experiment in the lung metastasis. Again, we first looked at the parenchymal cells, and you can nicely see that the metastases are very sensitive to rapamycin. And when you look at the fold change, it seems they're even more sensitive than the primary tumors, which is in line with the very first Western blot I showed you, where we always saw a higher mTOR phosphorylation in the metastasis versus primary tumor. The next thing what we did is we then silenced PHGDH. And as you remember, we saw decreased phosphorylation of mTOR targets up in PHGDH silencing. So now we would actually expect less sensitivity to rapamycin. And indeed, this is what we saw. So first of all, we saw the metastases were growing smaller because they had less growth signaling, but there was no additional effect of rapamycin. So in line with the idea that the serine biosynthesis pathway is important for mTOR activity. But we really wanted to be sure about this. So we said, let's also do the opposite. Let's overexpress PHGDH. So we use the cell line now that normally does not use the serine biosynthesis pathway. We overexpress PHGDH, which will activate the serine biosynthesis pathway and cytosolic alpha-ketoglutarate production. And then we ask, what happens if we now treat metastasis from those cancer cells with rapamycin? So first of all, we would expect that the parental metastases are rather insensitive to rapamycin, because the parental cells do not have an active serine biosynthesis pathway. And this is also what we observed, because we didn't see a significant decrease in metastasis area up in rapamycin treatment. But now when we overexpress PHGDH, we first of all saw that the metastases were growing larger, and that this increase in metastasis size was actually driven by mTOR activation, because when you give rapamycin, you bring actually back metastasis area to the control level. So based on these data, we actually can conclude that also in metastasizing cancer cells that activate proliferation, pyruvate is important, because it will activate the serine biosynthesis pathway, which in turn potentiates mTOR complex 1 signaling. In the consequence, we have larger tumor growth, but these metastases are also sensitive to rapamycin. When you now either inhibit MCT2 or PHGDH, so the serine biosynthesis pathway, there is less mTOR signaling, the metastases grow smaller, but the remaining metastases are rather insensitive to rapamycin. So I hope I have convinced you today that nutrient metabolism, and in particular proline and pyruvate metabolism, is important for metastasis formation, because I have shown you that cancer cells that just seed and colonize in a distant organ require pyruvate to remodel the extracellular matrix. They require proline to fuel their increased energy needs. And once the cancer cells have actually seeded and colonized the distant organ, pyruvate remains to be important, because it allows the cancer cells to potentiate mTOR complex 1 signaling. So with this, I would like to end, and I would like to thank my whole lab, all our fantastic collaborators, and our funding, and you for your attention. Thank you very much.